2017, PM Bhargava Oration Award 2017. So that's what he is, with over 85 publications in several prestigious national and international journals under his belt. His current research areas include understanding of endometrial receptivity and endometrial disorders, factors governing embryo implantation, genetics of sexual development and male infertility, COVID-19 and reproduction, and he's also the co-investigator of the pre-COVID registry, which collects information on pregnant women with COVID-19 and their babies in India. It's indeed a great privilege and honor to have him with us for this talk. Dr. Deepak Modi, we welcome you to our webinar. And to moderate the session, we have our senior consultant, reproductive and fetal medicine specialist, Dr. Koshi Cherian, sir. Sir is a doyen in the field, undergraduate and postgraduate training at Ludhiana. He went on to work as consultant at St. Stephen Hospital, New Delhi, from where he trained in IVF in Liverpool and went on to establish the IVF Center at St. Stephen's Hospital. He's also had his advanced USG training at Zagreb University and has been serving a lot of couples with reproductive and fetal medicine at St. Stephen's Hospital. And he's been our consultant ever since inception of our hospital. Welcome, Dr. Koshi Cherian, sir. Over to Dr. Koshi Cherian, sir, for the moderation.
Good evening, Dr. Jacob. Just thanks for your kind words. As I start this evening, this discussion, I wish to thank the Almighty Lord for this opportunity to interact with Dr. Deepak Modi and to learn from his vast experience in the field of reproductive biology. As you all know, in the year 1977, Steptoe and Edwards carried out a pioneering successful human embryo transfer, which resulted in the birth of the world's first IVF baby, Louis Brown, on the 25th July 1978 in Greater Manchester, UK. Since then, the ART has had big strides and has come a long way. Now we have better ovarian stimulation protocols. We have better hormonal assays. We have better ovarian induction methodologies. We have better... Hello? Am I audible?
Requesting all delegates to kindly mute your mics. Please mute your mics. Thank you. Please do continue, sir.
So we have better ovarian induction agents. We have better media. We have better reagents. We have better oocyte pickup techniques. We have much improved embryology laboratories. So, and we have blastocyst transfer, which is almost akin to the natural cycle. So, however, there is one problem that we all face as clinicians, that is the poor implantation rate, which has not kept pace with all the improvement that has been obtained in the development of embryo. So that's one of the impediments that we all as clinicians face. So this is attributed to the poor endometrial receptivity. Many studies have been done in this field. Many factors have been evaluated. Many reasons and blames have been put in. Endometrial thinning, endometrial echogenic thickening, vascularity, all these factors have been brought in. Ultrastructural endometrial epithelial changes have been studied. Various biochemical markers have been studied, but nothing so far has improved the receptivity of the endometrium. Now coming to the molecular approach, we have now advanced biotechnology, which will help us, and also in DNA microarrays, which helps us in simultaneously expressing the thousands of genes. And this can be employed or is being employed in evaluating the gene expression in the endometrium, especially in the menstrual cycles, also the menstrual cycles. We can study the gene expressions and also the effect of the stimulated cycles in endometrium. So while I understand that Dr. Deepak Modi is going to elaborate on HOMA10, which is been studied and have been found to be a factor in this endometrial receptivity. As we clinicians, we don't have much awareness of this and the clinical role of this. And today I expect Dr. Deepak Modi to elaborate and help us in this matter and explain all the intricacies of HOMA10. I understand that these are a lot of clusters, a lot of subgroups which are very difficult to be encoded or decoded. That itself will sort of for clinicians, as we clinicians, we find it difficult to understand the whole methodology, all intricacies of the fact. And today I'm glad that Dr. Deepak Modi is available with us to elaborate and explain and make us understand what is all this complicated genetic involvement in the endometrial receptivity. Thank you. Over to Dr. Deepak Modi.
Thank you, Professor Cherian. That's really very nice of you to set the stage for me, and it's going to really set the ball rolling for me, making my life easy, because you've really given a very nice overview of how from Bob Edwards and Siddharth Mukherjee that the IVF field has progressed today and what are the lacunae there. And at the outset, I'll also like to thank Jacob for really pushing me through to come here and give you guys a talk. It is indeed a pleasure, and it's nice seeing that now we have a nice crosstalk between clinicians and basic scientists. So what I'm going to elaborate to you for the next 45 minutes is how some research done in the laboratory, how some observations made in the clinics get us to do research in the laboratory, and how this research eventually translates back into the clinics and help people to have fertility. So without spending too much of time, I'm going to start my presentation. Let me share my screen, and I hope it is visible. Sorry, it's starting. Okay, so I come from ICMR Institute in Mumbai, and this is one of the only institutes in the country dedicated to studies in reproductive health. And as my CV has been read out, which says that our interest has been in a variety of subjects, which includes understanding causes of infertility, male infertility, genetic aspects of it, and more recently COVID-19. So if time permits, I'll spend like about 15 minutes trying to even tell you about what we have done in COVID-19 and how we are trying to address its effects in reproduction, but that's much later. Let's understand this aspect that infertility is a big problem. Although at one side we talk of population explosion in this country, infertility is on equal rise. So currently, the ICMR estimates that about 1 in 4 couples experience involuntary childlessness, and nearly 2% of couples in India are infertile. And that's a huge, huge, huge number. And most of these couples will opt for assisted reproduction for biological parenthood. But when these couples go for assisted reproduction, most of the embryos that are there fail to implant. They just do not sustain. Only 25% of the IVF done in this country or anywhere across the world are only resulting in live births, as 75% of the cases just either go unnoticed as pre-implantation losses or post-implantation losses. So I'm sure Dr. Cherian will agree with me that a holy grail of a ART center, that is Assisted Reproduction Technology Center, IVF center, is that all the patients that walk into the hospital must go home with a baby, and that's why they have come to the hospital. But when all the nice things are done, the reality is that less than 30% of them take home a baby. Almost 70 to 80% of them, despite repeated cycles, fail to have a biological parenthood. And what is the cause of such a thing? So for years, as Sir said, that we have now had from what Bob Edwards and Siddharth Mukherjee has started, where we had from today we have wonderful hormones, we have very beautiful media available, there are lovely culture systems available, there are very trained embryologists and IVF clinicians there. We can now study the embryo live in a culture dish. We can monitor every minute of the embryo growth. We can now even take biopsies from these single cell biopsies or multi-cell biopsies from these embryos, study the genetic profile. And we have paid a lot of effort has been put to improvise the IVF treatment. But come whatever that you do, most of the times the embryos fail to implant. So why do they fail to implant? One of the theory that tells us is that like a healthy seed cannot grow without a fertile soil, a healthy embryo is not sufficient to ensure pregnancy. The endometrium also has to be well prepared. So what is this well-prepared endometrium? And medical textbooks tell us that a blastocyst requires something called as a receptive endometrium for implantation. And how is this receptivity achieved? The ovary produces hormones like estrogen and progesterone sequentially, and a combined action of estrogen and progesterone during the mid-luteal phase endows a very specific phase in the endometrium, which is called as the window of receptivity. So if embryos come before that or after that, the window of receptivity is not achieved. And for years, we have all tried to understand how to get the window of receptivity in the best manner. What are the molecular features here? What are the things that's happening in the endometrium that allows implantation? And this is because we have spent so much of time is because of the assumption that the receptive endometrium is a passive tissue, which should allow embryo implantation. And for years, we have spent time, and currently there are very wonderful diagnostic technologies, which if you take a biopsy from a woman, and if you put it onto a microarray, it is going to tell you whether she's in the receptive phase or not in the receptive phase. Having said that, there is enough clinical data around which tells us that a receptive endometrium, measuring receptivity of the endometrium is not improving pregnancy rates. For example, several years ago, measurement of LIF in the endometrial fluid was thought to be a marker for receptivity, but if you have good amounts of LIF in the endometrium, but there's no correlation with pregnancy rates. Endometrial thickness, as Sir said, blood flow color by Doppler, these all we have applied and shown that there is a receptive endometrium, there is a good quality embryo, but the embryos just fail to implant. So the question that intrigued us was that despite a very good quality embryos which are being transferred into the endometrium, why does the embryos fail to implant? What is the cause of such implantation failure in these cases? So the first thing that we challenged was this hypothesis that a receptive endometrium is a passive tissue where the embryo has to just implant. Is it really true that if the receptive endometrium is there and if the embryo comes that it has to implant? Can something go wrong here? To address that, we use several model systems. So because it's very difficult to get human tissues and it's obviously not ethical when a woman is fertile or when the woman is infertile and we do expensive embryo transfers, and then I cannot go to Sir and tell him that, Sir, give me some endometrial biopsy because I want to study why there is no implantation, because we wanted to study after the receptive endometrium and the embryo is apposing onto the endometrium, when it is attaching to the endometrium, how is it invading inside the endometrium? We wanted to study these events because we thought that these events could go faulty in women with implantation failures. So for that, we use the mouse as a study model, we use monkeys as the study model, and sometimes we use human biopsy tissues or cell lines as a study model. So for all through my slides, I'll be using some of these symbols to let you know that from which study model is this data coming from. But the implantation process in the mouse, monkeys, and the humans is nearly similar, except that the timelines are slightly different. So several years ago, one of my students asked that, can anything happen to the endometrium just in presence of the embryo? So before that, I start with this. I'd like to tell you that from the pre-receptive, we have the receptive endometrium. Then the embryo starts coming here, and it is called as a pre-implantation stage. Then it goes to the apposition stage, and then the attachment stage. So what this student did was she took monkeys, she allowed them to mate, and she collected biopsies of the endometrium when the embryo was in a pre-implantation stage. So the panel that you see here on the left side are biopsies of the endometrium of monkeys, which were non-pregnant, but in the receptive stage. Here on the right side, these are biopsies from the endometrium of monkeys, which were allowed to mate. So these both are in the receptive stage, but the only difference between these two groups is there's a presence of a embryo here, which is yet not implanted. And here, look at the difference. This is histology. This is the luminal epithelium, which is very nice, flat and thin columnar epithelium. And here what you see is, look at the size of the epithelium. It is increased. It's become so huge. There's massive proliferation of the epithelium just by the mere presence of the embryo. Then we go to the molecular level. At the molecular level, our favorite protein is hoxatin. And we see that on the luminal epithelium, there is bundles of hoxatin sitting here. Don't worry about what does it mean, but try to remember this term hoxatin. This is a gene which is very crucial for receptivity. And I'm going to tell you more details about this gene a little later. But you can see hoxatin is, a lot of hoxatin is in the epithelium and there is in the stroma. But when there is presence of embryo, the hoxatin disappears from the epithelium and it is increased in the stroma. Another protein, MUC1, tons and tons in the receptive endometrium. But in the pre-implantation stage endometrium, MUC1 is reduced. Integrins, very little integrins in the receptive endometrium, but when there's a presence of the embryo, the expression of these integrins increase. So what does this tell us? That in presence of the embryo, there should be some changes happening into the endometrium. So we thought, let's explore this further. And we also did the studies in the mouse. So the mouse is a very beautiful system because we can time the pregnancies in them. So on the day when we put the animals for mating, the next day morning, we can see a vaginal plug. And if we take the animals 4 days after that in the morning, we can see the embryos nicely floating into the endometrial lumen, but it has yet not apposed. If we take this endometria after 12 hours, we can see the embryo is now nicely sitting and apposing onto the luminal epithelium, which is now embedded into the implantation cup. And by the next day evening, that is within 24 hours plus 12 hours, we can see that the embryo is now starting to invade into this endometrial bed. So we asked, what are the changes happening in the mouse endometrium during this time point? And this was one of our favorite experiments. This is called as oviductal glycoprotein 1 or OVGP1. So this is a protein we looked at for very random reasons. And here you see that in the pre-receptive stage, there is no OVGP1 in the endometrium. When the endometrium is in the receptive stage, there is no OVGP1. But as soon as the embryo starts apposing onto the endometrial, we see this beautiful red light here, which is the staining for the protein called as OVGP1. And loads of OVGP1 starts coming onto the endometrial epithelium, which is there on day 4 evening, day 5 morning, and when the embryo starts invading also, there is OVGP1. So this is one protein which is induced into the endometrial epithelium only in presence of embryo. It is not seen in the receptive stage. So my student asked that, is it necessary? How important is this protein OVGP1 in the endometrial epithelium? So we took human endometrial epithelial cells and we knocked down this protein. By knocking down, what I mean is that we reduced the levels of these proteins. So now we had something where we had normal levels of these proteins and something where the levels of this protein was terribly low. And we looked at integrins. Remember, integrins are proteins which are there on the endometrial epithelium to which the blastocyst is going to go and attach. Now, you see here that the expression of all these integrins which allows blastocyst attachment, the expression of these integrins is terribly reduced. She also looked at the levels of these on trophoblast adhesion. So we took trophoblast cells and tried to see how well they adhere to these endometrial epithelial cells. And if you lose OVGP1, there is reduction in trophoblast cell adhesion. Remember, trophoblast cells are from the embryo. And this is our wonderful data. So we also took data from women experiencing recurrent implantation failure, and we found that the expression of OVGP1 was terribly low. So these results together told us that in presence of the embryo in the endometrial epithelium, there is expression of OVGP1, and this embryo-driven OVGP1 is necessary for the embryo to attach correctly and then implant. And if the levels of these OVGP1 goes faulty, then there could be recurrent implantation failures. This is another protein, our favorite, hoxatin. In the receptive endometrium, we see lots and lots of hoxatin. You can see this beautiful red-colored protein sitting in the luminal epithelium, sitting in the stroma. But you see, as soon as the embryo is starting to appose here, this is the embryo sitting here. Of course, it's in the mouse. We cannot do the study in the humans. And you see, compare this to this. The expression of hoxatin is reduced dramatically. And you see here, here's a close-up, lots and lots of hoxatin in the endometrial epithelium, but the hoxatin is completely downregulated at the time point when the embryo has to start implanting. So we did a lot of these dynamic and kinetic studies. Let's not worry about it. But our results tell us that from pre-receptive to receptive, the levels of hoxatin go up. Then the levels of hoxatin reduce in the epithelium when the embryo implantation starts, but it is increased in the stroma. And when the embryo is to invade, it is reduced further. So what's happening here? So similarly, we have done lots and lots of protein expressions here. So what we have reviewed this work, and here we show that in the presence of the embryo signals the receptive endometrium. So the receptive endometrium has to get modulated by signals from the embryo, where the signals from the embryo allows proliferation of the epithelial cells in a very low. Am I audible?
Hello.
Yes, you are. Yes, you are.
The embryo allows proliferation of the epithelial cells. It allows decidualization of the stroma. It increases expression of a lot of genes. It reduces expression of some of the genes. And there is also changes in the angiogenesis in the stroma. So before the embryo implants, the endometrium sets a round of preparation. So this is, you can imagine, it's analogous to a guest coming at our home. So if some guests have to come at our house, the house needs to be prepared accordingly. So even before the embryo enters into the endometrium, it calls it and tells it, hello, I'm here, and prepare yourself to welcome me. So the way we look at this is that the embryo prepares the endometrium prior to implantation so that it is ready. And it is possible that some of these crosstalks may go faulty in women with recurrent implantation failure. So I'll move slightly ahead here. What's happened to the slide? So the myth, the earlier hypothesis is a myth that the receptive endometrium is a passive tissue that should allow embryo implantation. The truth is that the receptive endometrium is modulated by the incoming embryo, and this is required for implantation. And if this modulation or crosstalk goes wrong, you could have implantation failure. So now once the embryo is apposed, it has to attach onto the endometrium and then start invading. Now remember, I told you that there is this luminal epithelium on the endometrium, there is the stroma, and the embryo has to breach this luminal epithelium. Now remember, the luminal epithelium is a very thick tissue, it's a very tight tissue where the cells are very tightly packed with each other and nothing can go in, nothing can come out. It's like a wall of a fortress. Now the embryo has to breach this wall of the fortress, and how this is achieved was one of the questions that my lab is still trying to answer, and unfortunately we understand very little of this process. But several years ago, a hypothesis was put forward. So what they did was they took mice, they overectomized that, and then injected them with only estrogen, and the embryos could not implant. They injected only with progesterone, embryos could not implant. When they injected with estrogen plus progesterone, the embryo implanted. And if you just scratch the luminal epithelium, even in absence of hormone priming, the embryo could implant. So at that time, for years, the researchers thought that at the receptive stage, the endometrial epithelium is a very weak tissue so that the cells just dissociate and fall apart so that the embryo can implant. So we looked at this and we asked whether it's really a weak tissue. And my student looked at this protein called as E-Cad. Endometrial E-Cad is a epithelial cells protein which keeps this epithelial cells together in a very tight form. And here you see that as compared to the proliferative phase, in the receptive phase, there is tons and tons of E-Cad which is keeping the endometrial epithelium intact. And you can see here so beautifully, all the cells are touching each other and keeping like a very tight zipper ziply attached to the cell, so nothing can go here. But as soon as the embryo started implanting, it apposed onto the epithelium, some magic happens and the luminal epithelium starts reducing its E-Cadarin. And this is one of my favorite slides. You can see this beautiful embryo trying to oppose into the luminal epithelium. This is the side at which it is opposing. And you can see there is no E-Cadarin on this side, and there is lots of E-Cadarin on the opposite side where the embryo is not edged to implant. So what did this tell us? That the receptive endometrium does not have a weak epithelium. In contrast, this is a very strong epithelium, and the embryo somehow has to chop these E-Cadarin so that the epithelium becomes weak and the cells try to move apart. So then how do these cells move apart? So we started looking at literature, and something told us in cancer where the cells fall apart for metastasis. So that is the process called as epithelial to mesenchymal transition. So we asked whether epithelial to mesenchymal transition causes, is one of the events associated with embryo implantation. We looked at lots of proteins. We look at E-Cadarin and N-Cadarin. So epithelial cells should have high E-Cadarin, low N-Cadarin, and mesenchymal cells, which are highly motile and moving, have low E-Cadarin, high N-Cadarin. So my students said that does loss of Hoxetan at the time of implantation cause EMT? Let's not worry much about it, but let's look at whether EMT does occur at the time of implantation. And here what we saw was that yes, the epithelial cadarin is reduced significantly in the embryo, and at the same time there is increase in N-cadarin, telling us that the cells which were very tightly packed with each other can now slowly move apart. So this was magical event which was happening. And I told you that already when the embryo is implanting, there is loss of Hoxetan. So we asked that could these cells be moving? So if the embryo has to now sit on the epithelium and the epithelium has to move apart for the embryo to get inside, are these cells mobile? Now, these are very difficult experiments to do when we are talking at the site of implantation, which is a very delicate tissue, and we really can't go inside the mouse and try to see what's happening there. So we do some in vitro experiments. So she took human endometrial epithelial cells and she said, okay, let me deliberately knock down Hoxetan. So she removed the Hoxetan from the epithelial cells, and this upper panel is the data where Hoxetan is in normal amounts, and the lower panel is the data where Hoxetan is in reduced amounts. And you see that the expression of E-Cadarin is reduced simply by the reduction of Hoxetan. And N-Cadarin, which is never expressed by these endometrial epithelial cells, suddenly starts gaining N-Cadarin. Remember, there is no embryo here. So these experiments told us that yes, in absence of Hoxetan, the epithelial cells become mesenchymal-like. Now remember, I told you that the cells have to move. So we did something called as live cell imaging. We put the cells under the microscope, both of these normal Hoxetan and low Hoxetan. We put them under the microscope and tried to see how they are moving. Try to pay attention here. These are control cells. These are when there is normal Hoxetan, and you can see there's dots. There is barely any movement. This is over 24 hours. You can see some cells are moving here and there, but you will see the magic when you see cells with reduced Hoxetan. Look at the speed, the same time, the time intervals are the same. I'll play both the movies together. You can see so beautifully these cells are moving. They all were running helter-skelter in the dish, whereas the normal cells are like all tightly adhered in its place. So this experiment told us that if we lose Hoxetan, the epithelial cells somehow become very mobile and start moving. So currently the story so far for our understanding of implantation is that before implantation, when the endometrium is receptive, the embryo comes in, signals the endometrium, and makes a lot of changes in the luminal epithelium and the stroma, such that it is able to tightly adhere first onto the endometrial epithelium. Once it adheres, there is loss of Hoxetan, there is gain of OVGP1, and there is gain of N-Cadarin and loss of E-Cadarin. This will now make the epithelium mobile. and the epithelial cells would just simply move apart beautifully, and now the embryo can invade. Now you can imagine yourself sitting there in the endometrium, and you can watch these events happening. That first the embryo comes in, there's a tight endometrial epithelium, the embryo comes in, starts and sits here, and then slowly the cells move apart and the embryo gets inside. So these are very, very delicate processes, very intricate processes which are happening at the time of embryo implantation. So now how does it matter in the clinic? We ask that what is it that's coming from the embryo that's going to make out these changes? How are these changes induced? So one of our favorite molecules, which we all know the embryo produces, is chorionic gonadotropin. The trophoblast cells of the trophectodermal cells of the embryo produces HCG, and this HCG could act onto the endometrial epithelium and the stroma to induce a whole lot of changes. So we asked whether the OVGP1, which we saw, the increase in OVGP1, which we saw in presence of the embryo, could it be induced by HCG? And yes, we could show that estrogen and progesterone, so this is estrogen and progesterone alone, which was not sufficient to induce OVGP1, but you look at this estrogen plus progesterone plus HCG, and you can see that HCG increases expression of OVGP1 significantly. And if we treat the cells with HCG alone also, it was sufficient enough to drive HCG expression in the endometrial epithelium. So these results together told us that the embryo secretes HCG. The HCG is sensed by the endometrial epithelium. It starts producing OVGP1, which then increases expression of integrins for the embryo to oppose. And there are several other papers, others in the world. So this is a data from my collaborators' lab in the US. And here what they did was, instead of doing these studies in the lab dish, they did it in the vivo. So what they did was they took women who were volunteers, they injected HCG into the endometrium when they were in the receptive phase, and they studied the receptivity, and they said that there is no major change in the receptivity, but in the women in whom the HCG was injected, there was a massive increase in estrogen receptor expression, progesterone receptor expression, and there was increase in expression of notch ligands, C3, so on and so forth. So this told us that the endometrium, which is now out of sync, so in many infertile women, the endometrium is out of sync. It is in the receptive phase, but it's not a synchronous receptive phase. HCG, which is produced by the embryo, gets them in the synchronous phase, and that should approve implantation. So then there was a time for this litmus test. So what this paper concludes is that HCG induces changes in the uterine stroma in preparation to implantation. So now the litmus test. Now, sir will ask me that all this is great. This is all in your mouse and your monkeys and your cells in the dish and some in the men injected with it. But what happens to pregnancy rates? Can we improve pregnancy rates by coaxing the endometrium? So the hypothesis that we put here was, so of course, this is not done by our lab because we don't have ethics approval to do these kinds of work, but these are done in my collaborators' lab, some of my friends' lab abroad. So you can imagine that what we started in 2005 of showing that there is HCG can induce into the endometrium, there are studies now done where they've injected HCG into the endometrial lumen, and after that, they've put the embryo and tried to see, do these women become pregnant? Lots and lots of studies are available. This is one such systematic review and a Cochrane review, which was done last year. And very beautiful data where they have shown that if you instill HCG into the endometrium, just prior to implantation, the endometrium starts thinking that the embryo should be coming here. It prepares itself for receptivity. And here you see that there is a massive increase in pregnancy rates, and the relative risk ratios, the RRs calculated is nearly 1.5. That means that for every 10 women without HCG treatment, 3 additional women would become pregnant. So if the implantation rate in today's scenario, when we do not do HCG injections, is 3 on 10, if by implementing HCG, nearly 5 to 7 on 10 is going to become pregnant if you start instilling HCG into the endometrium. However, I must put a caution here that there are a lot of variability in this data. The data quality is not very great, but it is very promising. So it is very satisfying for me to sit here and talk that all the studies which we started almost 15 years ago from now, that we showed that the embryo can induce changes into the endometrium. At the end of 15 years now, we have clinical data to show that embryo-driven changes can occur into the endometrium, and such changes can help to improve, and such changes are necessary for implantation to occur. And if these changes are not occurring in women, then they can have pregnancy failures, and you can inject HCG in the women, and you can improve pregnancy rates. So this is the take-home message from my side, that the blastocyst, which is there, produces a lot of embryonic signals like HCG and other molecules, which induces a lot of morphological changes into the endometrium. And these morphological changes are necessary to are necessary for the embryo to implant. And conversely, such changes into the endometrium also tells the blastocyst to increase its adhesion rate, to increase its invasiveness, and also to do immunomodulation. So why should this process occur? Why should such a process of the embryo-driven changes occur onto the endometrium? So this is what we think is because the endometrium is like a biosensor. Remember, human pregnancy is a very, very labor-intensive process. It's an energy investing process. And we know that a lot of embryos may morphologically look nice, but they may not be developmentally competent. So the endometrium needs to make a choice. It cannot allow every embryo to implant, because if the embryo quality is poor, then the pregnancy will discontinue much later. So the mother invests a lot of energy into the to induce pregnancy, and if the embryo is not sustainable, then this process cannot be achieved. So we think that such a embryo-driven changes into the endometrium actually are for a embryo crosstalk. So when there is a healthy embryo coming into the endometrium, right kind of changes happen into the endometrium, and the embryo implantation and invasion can happen. But if the embryo quality is poor, it could have a metabolic overdrive, it could be genetically unhealthy, it could be metabolically unhealthy, it could be morphologically unhealthy. And such embryos, if they come into the endometrium will now sense these signals, thinking that something is not right with the embryo which is coming inside, and it terminates its receptivity and goes into menstruation. So here is what we propose, that this is something which we have discovered, that the embryo implantation is rather a three-step process. The first step is hormone-driven changes into the endometrium to make it receptive. Remember, receptivity is necessary, but it is not sufficient. What is the next step that is necessary is that on a receptive endometrium, the embryo has to make second round of changes. And when the second round of changes happen, that the embryo can oppose an implant, and there's decidual transformation. And once the embryo opposes and implants, epithelial to mesenchymal transition has to happen for completion of process of implantation. So these are two new steps which we have discovered in the process of embryo implantation. And we're very proud to say that our basic research which has happened is now really helping clinicians to give their patients an option to have better pregnancy rates. So what are the endometrial determinants of a successful pregnancy? We think that the endometrium, first of all, has to be reasonably receptive. So if it is poorly receptive, then there could be implantation. There would be no implantation, or there could be implantation failures. But also at the same time, this embryo-driven changes in the endometrium allow a selector activity. It allows selection of the embryos, and such a selection of the embryo is necessary to reach successful pregnancy. And a very good balance between the receptor activity and the selector activity is going to help in initiation of pregnancy. And if there is a disbalance, if there is excessive receptor activity or poor selector activity, such patients will have often miscarriages. or if they have too much of selector activity. Suppose in a woman there is too much of selectivity, she does not allow any embryo to implant, then she will manifest as infertility. So miscarriages and infertility could be opposites ends of the same spectrum, where if the embryo is unable to decide the quality of the embryo, every embryo that comes in will implant, but the embryo will be developmentally incompetent. It will culminate into a miscarriage. And at the other time, it is not allowing any embryo to implant, and it is going to manifest as infertility. And if there is again a problem in the selector activity, the receptivity is good, it could also manifest as recurrent implantation failure. And we have some data to show that if these selector receptor activity also goes altered, they could manifest as preeclampsia, IUGR, or preterm birth. So we think that initiation of pregnancy is the key for initiation. The implantation process is the key, and the implantation process is governed by a receptor and the selector activity. Now, here is a major problem. So what we call this is embryo implantation combat, where we see that the embryo is there to implant and the endometrium is like a opposing pole. And this combat has to occur, and then the embryo and the endometrium is to decide whether it wants this embryo to implant. And only then a successful pregnancy will occur. Now, here is a challenge. The challenge comes is that if you will ask me that now, how do I measure the selector activity of the endometrium? Here is a woman who is infertile. She has recurrent implantation failures, 5-6 rounds of IVF done, and she is not getting pregnant. I will say that it looks like that, oh yeah, she might have a high selector activity. But now if you ask me that is this woman have an appropriate selector activity, or she is a poor or high selector activity? We don't have toolkits. We don't know how to measure that. We only currently know how to measure receptivity. We don't know how to measure selector activity. And so for now, for people like me and for people like Sir, it's time to crosstalk. We clinicians, basic scientists, researchers have to now crosstalk and get together. Embryologists, IVF practitioners and scientists have to crosstalk now, and we have to figure out how do we measure this, because currently we don't have any toolkits to measure selector activity of the endometrium. But once we are able to move to that step, so that's the next step my lab is going to get into, that we are trying to see, can we develop markers to assess selector activity of the endometrium so that we can help clinicians to have better pregnancy rates. I'm not put here, but there is a paper which we have published. We call it as Embryo Implantation, Love in Times of War. Here is where we have summarized this entire process of embryo endometrial crosstalk, and to explaining it use a very simple analogy that how this crosstalk is very crucial for successful pregnancy. I have 10 minutes, right?
No problem, Dr. Deepak. You can take your time. No problem.
Yes, I would like to just briefly run through what we have done in COVID-19 and pregnancy. We are in these times of pandemic, and somewhere in March, where there was this immense lockdown put into the country, across the country. And we realized that there's so many people who are getting infected, and anticipating that more would get infected. Our major concern was that can COVID-19 affect pregnancy? And so this is completely different. My gears are totally switched here. So some of you will take some time to adjust to it. But I thought this is very close to my heart, and this is something which we have initiated recently. And I thought I should share with all clinicians who are here, because it's very crucial that we try to address this problem as quickly as possible. So we know that it's one of the largest pandemics that we have come through. Millions and millions of people are affected, and obviously pregnant women are affected. So when we started, when the pandemic started and it was slowly percolating across the world, a lot of questions started arising. And to us in our institute and through ICMR, we had a lot of questions which were being asked to us persistently, that can COVID-19 so we know that it is caused by the new coronavirus, SARS-CoV-2, but can it infect the embryos? If a woman gets pregnant, so now the IVF centers were the first ones to call me and ask me that in this pandemic, should we have the centers open? If they're open, if there is somebody with an infection coming in my center, is it going to affect the fetus? Is it going to affect the embryo? Can we offer IVF during this pandemic? Because are there any ill effects of this virus during pregnancy, during early implantation? And Is it going to adversely affect, is the SARS-CoV-2 going to adversely affect pregnancy? So we had many such questions being bombarded to us, and we thought we'll have to address these questions quite systematically. So to do that, what we did was, so this was all thanks to my students who worked really very hard during the lockdown. So by the time when by March, we had known reasonably good about the virus. We knew that SARS-CoV-2 requires ACE2 to bind onto the cell surface. Then the virus is going to get cleaved by this protein called as TMPRSS2, and this will allow virus activation. So the SARS-CoV-2 is going to get cleaved by the TMPRSS2 to allow efficient binding. And then the viral genome is going to again get released inside the cells. And then here it's going to get under replication. And after replication, it has to bud out. And there is another receptor called as basigen. So we asked that if the embryos have to get affected by SARS-CoV-2, they should express these receptors. And if they don't express these receptors, the embryo should be safe. So my students we started wondering, how do we address this? No IVF clinic is going to give us live human embryos, and we cannot have the virus in hand to start infecting anything and everything we get. But then my students were quite smart here. They said that, look, there is a lot of data available here. So there is single cell RNA-seq data available, where every cell of the human embryo, the RNA content, the RNA genes, the genes which are expressed inside every cell of the human embryo is mapped, and it's there in the database. So my students downloaded this database, single cell RNA-seq database of human embryos, and they started looking for the SARS-CoV-2 receptors. I'm sorry, this is a little smaller handwriting here, but you can see that in the zygote and the 4-cell embryos, there is lots of ACE2. The expression of ACE2 reduces, and then the expression of ACE2 increases suddenly into the embryo when it is ready to implant. And this is in the trophectoderm when the embryo is at the blastocyst stage. You can see there is tons and tons of SARS-CoV-2. There's lots of TMPRSS2. There's a lot of BSG, and most of the proteins which were required for the virus binding and entry are present on the human embryo. And this was very, very surprising to us. We never thought that the early human embryos would express something like ACE2. And then my students asked that, okay, even if the protein, even if the RNA is present, and if the virus, the receptor is there, the virus has to get inside, replicate, and then bud outside. So if this machinery is absent, then the embryo should be safe. Something binding doesn't mean it is good enough for infectivity. So they looked for genes which are required for viral replication, and lo and behold, you see here, these are all some 15 to 25 genes which are required for viral replication. These are host genes, and almost all of these genes are very heavily expressed in the trophectoderm of day 5 and day 6 blastocysts. So at the blastocyst, as the trophectoderm not only expresses the receptors for SARS-CoV-2, but they also express the viral replication machinery and the viral budding machinery. So what these results tell us, that the embryos, the early developing human embryos, have ACE2, they have TMPRSS2, they have the necessary machinery for a virus to enter inside, they have the necessary machinery for the virus to replicate, and it can even bud outside. So the entire infectivity cycle is present in the virus. So if the embryos experience SARS-CoV-2, it could be potentially damaging. So one may ask, that how does SARS-CoV-2 can come in an IVF lab? There are many such sources. For example, if there's an infected patient, infected lab, or a clinical personnel, and remember 40% are asymptomatic in the first 5 days of infection. And today we know that most patients are asymptomatic, and they can generate aerosols, and they can put to other susceptible individuals. Remember, these aerosols can sit on your incubator, that can sit on your flask, they can sit on your plastic wares, glasswares, they can come on contaminated surfaces in your operation theater. And we have clean evidence now currently in the literature which tells us that SARS-CoV-2 can survive for very long time, almost 3 to 7 days on such surfaces. And it can now get and contact your lab and clinical personnel, or it could be an indirect contact, and such a person can now start infecting embryos. So it's going to get into a vicious cycle. So those who are already practicing IVF today, I would really urge to think that your embryo is very susceptible to SARS-CoV-2 infection, and it is very necessary to not just test the clinician and the incoming patient, but the staff, the laboratory staff, the one who is handling the embryos, the one who is handling the gametes, have to be continuously tested so that there is no evidence, so that the SARS-CoV-2 does not get into your lab and infect the embryos. Also, the good news is that the embryos have very little amount of ACE2 and the machinery from the 4-cell to 8-cell stage, and there is a thick zona pellucida covering. So if you want to practice IVF, do practice it, but then freeze the embryos by day 3 or day 3. Do not allow them to go to day 5 when there is lots of SARS-CoV-2 receptors. By day 3, freezing and day 3 transfers can be safely done, and the embryos can be protected. So you can change your practice currently, and also you can test your individuals so that you do not have such breakthrough infections. If you want to know more details, there is this paper in archives available, or it's free online. It is called as Expression of SARS-CoV-2 Receptors and Spike Proteins in Developing Human Embryos. You can read this paper and feel free to contact me. I'll be happy to answer your questions. The last question that we were addressing was SARS-CoV-2 in the placenta. The same question my students asked, that can the placenta also get infected by SARS-CoV-2? Why placenta? We thought because even if the mother is infected, the placenta is a nice barrier which does not allow any infectious agents to cross through here. And our expectation was that the placenta should not contain any SARS-CoV-2 receptors. If at all it contains, then there would be a very strong risk of mother-to-child transmission. So again, we took single-cell RNA-seq data. We tried to deconvolute it. I'm not going to go to the technical parts of it, but here is the data. We found that the syncytial trophoblast of the first trimester placenta abundantly expresses ACE2. Even the extravillous trophoblast expresses ACE2. Here we show that both ACE2 and TMPRSS2 are co-expressed in almost all the cell types of the human placenta in the first and the second trimester. And the viral replication machinery and the viral budding machinery are abundantly expressed in the syncytial trophoblast and the extravillous trophoblast of the first and the second trimester placenta. And so we show that there is a lot of BSG expression and there is enough ACE2 expression for the placenta to get infected by SARS-CoV-2. So then obviously when we published this work, this was published in Frontiers in Cellular and Frontiers in Cellular and Developmental Biology. And when we published this work, a lot of people asked, can it really infect? You just show that the RNA is there, but is it really infected? And if the placenta gets infected, can it transmit to the baby? And luckily around that time, we had this one very interesting case. This is the last 2 slides that I have. This woman was pregnant and she was at 7 weeks. She came to one of our collaborators' hospital, and she got herself, pregnancy was confirmed, and she was asked to come back after a couple of weeks for a routine follow-up. But unfortunately, she got infected by SARS-CoV-2 within a week. But this was asymptomatic, absolutely uneventful, and she recovered very nicely. And she came back to the clinic at 13 weeks of gestation, so 7. And 5 weeks later, she came for pregnancy. She came to the clinic for an ultrasound. And the ultrasound report is here. And you can see that it was not normal. There was no fetal cardiac activity. There was no blood in the fetal heart. And you can see that the membranes, there was a lot of subcutaneous edema. You can see there's a lot of edema here, and there was pleural effusion. And the diagnosis, the radiologist gave us a diagnosis of hydrops fetalis and fetal demise. Now, this was very intriguing. She's infected at 7, she's infected at 8 weeks. 5 weeks later, she comes. So the first thing that the doctors did was tested her for SARS-CoV-2, and she was negative. So she recovered completely from SARS-CoV-2, but her fetus had died. And now we had to ask whether there is any association of the mother and the fetal, mother's infection and the fetal demise. So what we did was we first did RT-PCR for SARS-CoV-2. So a nasopharyngeal swab at the first time was positive, but it was negative on the day when she underwent termination of pregnancy. But when we collected the placental tissue and the placental tissue medium, so we incubated this placental villi for some time in the viral transport medium, and we found that the viral transport medium, so imagine an infection is at 8 weeks, recovery within 5 to 7 days, and even 3 to 4, even 3 weeks later, her placental tissue had lots and lots of SARS-CoV-2 RNA. The SARS-CoV-2 RNA was also found in the amniotic fluid. It was unprecedented. This is the first time ever we have reported in the world that the first trimester infection, completely asymptomatic infection, and now you see the placenta was infected with SARS-CoV-2. But then we were not happy just looking at the RNA, because RNA can just float around and it need not be inside the cells. So my very bright student, Anshul, did immunofluorescence. So we took antibodies against SARS-CoV-2, which binds to SARS-CoV-2, and we did immunofluorescence on placental sections. And here is what you see. This is you're seeing for the virus for the first time in the world in the first trimester villi. You can see this beautiful, this is the first trimester placental villi. And you can see the villi, the syncytotrophoblast layer of the villi. You can see there is lots and lots of SARS-CoV-2 sitting here in the first trimester villi. So where we predicted there is presence of receptor, here we show that there is not just presence of receptor, but SARS-CoV-2 can actually enter inside the placental villi and happily replicate there, and it replicated there for 2 full weeks. And not just in the placenta, but it crossed the transplacental barrier, it got into the amniotic fluid, and it even infected the fetal membrane. You can see here there's a fetal membrane tissue, and in the fetal membranes also we found a lot of SARS-CoV-2 sitting there. So these results told us that there is a strong possibility of vertical transmission of SARS-CoV-2 from mother to child. Now remember, this is a respiratory virus. Unlike Zika virus, which is a blood-borne virus and it can straight go to placenta, this is a respiratory virus. So it will not be seen in all the women, but in a subset of women, if they're infected, the SARS-CoV-2 can get shed into the plasma. And if the SARS-CoV-2 from the lung gets shedded into the bloodstream, from the bloodstream, it can go into the placenta. Remember, in the placenta, I've told you that these are the syncytotrophoblast which have the receptors. The virus can sit onto the syncytial trophoblast cells, enter inside, happily proliferate, and it can now spread infection inside the villus, and through the fetal capillaries, it can go and infect the fetus. So here is what we show, that from the mother's infection, it can pass on the SARS-CoV-2 to the baby, and the baby can be infected. In our case, at least, we think that the baby died, the baby developed fetal hydrops, and the baby died is strongly because of the SARS-CoV-2 infection. Of course, it's a single case report, and therefore there were many other questions that came up through this, that are pregnant women more likely to develop COVID-19 related complications? How common is this vertical transmission? What are the presentation and the phenotypes of women with COVID-19 in India? And my colleague, Dr. Rahul Gajbhige, has started this Preg-COVID registry, in which I'm one of the co-investigators. So this is a registry. This is a hospital-based registry of pregnant women with COVID-19. Here we have about 18 medical colleges. So this is currently only operational in Maharashtra, and we are having requests from other parts of the country. But from Maharashtra, we have data from 18 medical colleges who give us the data of every pregnant woman that comes in there, and if she's SARS-CoV-2 positive or negative, and what are her symptomology, what was her clinical course, and what was her pregnancy outcome. Until October 2020, we have already registered 5,000 women in this registry. There's data of 3,000 infants and 2,500 mother-infant diets. Data is very available, but currently it's not yet analyzed, so I cannot tell you what are the outcomes. But some of the early outcomes we have shown here is that almost 90% of the pregnant women who are infected with COVID-19 are going to be asymptomatic. So whenever there is a pregnant woman, people thought that pregnant women should be more symptomatic. Here it's becoming reversed, that they are asymptomatic. But we know that and we have published this paper very recently, and we also see that there is a higher incidence of preterm births, there's a higher incidence of preeclampsia, and there is an increased rates of maternal deaths. So currently this data is under review, but if you want to know more about Preg-COVID registry, do visit our website. We'll be happy, and please feel free to email me or Dr. Rahul. We'll be happy to give some information. So the take-home message for the COVID part is that early human embryos are permissive to SARS-CoV-2 infection. So if you are an IVF person practicing IVF, be very, very careful because there could be a strong embryo damage. Again, like the human embryos, the human placenta has the necessary machinery for SARS-CoV-2 infection and replication. And we have shown you that the SARS-CoV-2 can really infect the human placenta and mother-to-child transmission can occur. Most pregnant women, as I've told you, are SARS-CoV are asymptomatic, but there is a higher incidence of pregnancy-related complications. So this virus doesn't seem to be as straightforward as it appears to be. There are a lot of backends to that, and we need to do more studies in pregnant women so that we can protect our future generations. And I think I will close here. I'm really thankful to my students. Today, the data that I've presented was generated by wonderful student Nancy, and all the work on placenta was done also by Nancy and Anshul. Anshul was also instrumental in doing the vertical transmission work. And we have a lot of collaborators for all the work that we currently do. And I thank ICMR, Department of Biotechnology, Department of Science and Technology, and CSIR for all the money that we need to do this work. And I hope you have enjoyed this presentation. Once again, thank you for being here, and I'll be happy to take questions.
Thank you so much, Dr. Deepak, for that amazing presentation. Really grateful for that. Over to Dr. Koshi Cherian, sir, for the moderation.
Hello, am I audible now?
Yes, sir,
Yes,
you
sir.
are.
Okay. Hello.
Yes, sir, you are.
Yeah, okay. So let me thank Dr. Deepak Modi for such an extensive presentation and also making it so clear for all of us. Of course, it's a very difficult and sort of very difficult topic to understand, especially for clinicians like us who are not involved in much of research work. But one thing which intrigued me is, in a normal pregnancy, what happens is when the embryo starts developing in the fallopian tube, the signal is already given to the endometrium and beta HCG starts rising. And also extensing corpus luteum should support, and progesterone also starts rising. That's the normal physiology which we understand. And in ART, what we do always give a trigger, the HCG, before the pickup. And also after the pickup, after the embryo transfer, we give HCG. So in additional HCG to the endometrial cavity is something new to me, which I didn't practice, and I'm still doubtful about the additional benefit of that HCG. But as you said, probably it would be, but is it statistically proved that it is helpful or not?
Yes, so I agree with you that in the natural cycle through the fallopian tube into the endometrium, there is enough HCG produced by the embryo, so pregnancy should occur. But in an IVF cycle, we must remember that we do a ET either on day 3 or on day 5. Now, when we do a ET on day 5, we are giving very little time to the endometrium to even sense the presence of the embryo. The trigger, that is HCG trigger, which is given is way early. That is for ovulation. By the time the endometrium has not developed any receptors for HCG. And the HCG support which you give is much later. It's not in the window of implantation where the HCG is actually required. So there are lots of studies there, and if you do a day 3 transfer with 500 IU of HCG, the pregnancy rates significantly improve, and the odds ratio is nearly 1.5 to 1.8. So definitely, but there is still a lot of gray area here because the patient selection criteria has been very different across studies. But we are convinced that it does happen, and I'm sure more trials are being conducted so that we can improve pregnancy rates. But definitely, it's during the window of implantation where all these changes have to happen. It cannot be much before that or much after that.
Thanks, Dr. Deepak, thank you once again. And if anyone else has any questions and queries, this is the time to ask. Anybody? Dr. Jacob? Is there anybody who wish to ask Dr.
Yeah, we are waiting for the questions to come in. There is an interesting question through WhatsApp from one of the students. Sir, we are currently pursuing medicine undergraduation. So how do we branch out into core research of this sort, which you have pursued? Can you throw some light on that?
Yeah, sure. So there are a lot of opportunities, especially for medical students, to pursue research in this country. First of all, ICMR does give a lot of fellowships to medical students. Like there is STS fellowship, which is a small amount, but a significant amount to pursue dissertations and carry out research in institutes. On the over and above that, once you complete your MBBS in ICMR, there are a lot of posts available only for medical candidates. So if you are a medical student and you are interested in research, you can get into one of those ICMR institutes as a scientist here, and you can pursue your research. Over and above that, you can pursue PhD. I mean, post medicine, you can do a PhD in one of those research institutes and then become a scientist in any of the labs in this country. So that's a technical way of going it. But research is something which I suggest is that it's for people. So I always tell clinicians that research and clinical practice is no different. Every day, every single patient that you see and diagnose is what you're doing is research. You're trying to get in new information from the patient. And all what we do is we collect all this information and put it up as a story. So I love this quote from Siddharth Mukherjee, who says that doing science is like stories. Patients tell stories to talk about diseases. Doctors tell stories to understand the diseases, and science tells the stories to explain diseases. So if you like to tell stories, if you like to interpret stories, then definitely research is for you. And I'm sure you guys do it every day. So it's fantastic. It's fun. And definitely you can pursue a lot of avenues in this country for medical students.
Thank you. Thank you so much, Dr. Deepak. We have our senior pediatrician, Dr. Elizabeth Verkey, online. So, ma'am, would you like to come in with your thoughts? All right. So, yeah, we have one. Yes.
I think there's a question in the chat window that says, does COVID-19 cause problems in second or third trimester of pregnancy? Definitely yes. From our pre-COVID registry, we do have data that in the first and the second trimester, it apparently looks very, very detrimental to the pregnancy. Third trimester also, there is infection, and infections in the third trimester are associated with higher risk of preeclampsia and preterm births. So definitely there is adverse effect of pregnancy throughout gestation, but at present we don't know which gestation age is more prone to problems and what are the manifestations same throughout gestation. That I don't know the answer because we don't know that information yet, but definitely yes, throughout across gestation, COVID-19 does have adverse effects.
Thank you, Dr. Deepak. One more question is that you mentioned quite a lot of protein molecules which are involved in implantation and more expression of certain molecules and lesser expression of certain other molecules. So is there any chance in future if monoclonal antibodies would play a role in enhancing fertility of prospective mothers?
Right. So this has been one of the thoughts for quite some time. So in fact, rather than enhancing, at some point our lab was interested in figuring out if we could make antibodies against some of these receptivity proteins and can they act as contraception. And there is definitive evidence towards that, but the problem is in most of these cases delivery. Getting it into the endometrium is very, very invasive process, and coupling it with embryo transfers and these processes is very, very challenging. And sustenance of those antibodies into endometrium is another issue, because at that point of time, the endometrium wants to clear off all the immune kind of molecules so that it's immunologically privileged. So it becomes challenging. I don't have a straight answer to your question, but there is a possibility that it could be done. And there are people who are trying to develop such not just monoclonal antibodies, but therapeutic molecules which can improve receptivity.
Thank you, Dr. Deepak. Is there any data about exposure to HCG, whether they pose any excessive risk of malignancy in terms of ovarian or breast cancer?
As far as we know, that these cancers produce HCG by themselves, but whether they cause cancers is not known. Giving HCG in clinical is a very routine thing in clinical practice. I don't think it enhances risk of any of these cancers, because as far as I understand, pregnancy is one of the events which reduces the chances of breast cancer. And what we speak about when we talk about giving HCG into the endometrium, these doses are ultra small. They're just less than 500 IU, or probably even slightly lesser than that. So these are not going to induce those kinds of changes which would lead to malignancy.
But one thing about ART and multiple hyperstimulation cycles, there are those cycles with the multiple hyperstimulation cycles and multiple oocyte aspirations and rupture and puncturing of the ovaries can, to some extent, lead to malignancy of the ovary. That is shown in many studies.
Yes, so
that's why people discourage.
Yeah, yes, go ahead. But I don't think that's the effect of HCG. That's most likely because of the fibrosis which happens because of those hyperstimulations.
That's right. It's not HCG. It is the hyperstimulation cycle.
So, if there are no more questions, I think
there is one. Why does obesity reduce the possibility of conception? A very nice question, and this has been one of the observations, that obese women somehow have lower conception rates. Most likely it's because of a metabolic disbalance. Because of obesity, there is increased levels of leptin, and there is increased metabolic turnover, which somehow disrupts endometrial receptivity. And because of that, there is a change in the receptivity, and therefore there is a poor implantation rate. We don't know about in women, but there is a colleague of mine at the institute who does work on obesity in males and the effect on conception. And what she shows is that obese males, there are a lot of epigenetic changes happening in the sperm because of obesity, and that has effects on embryo development. So most likely, even in the female, same things might occur, where obesity may change, where may have epigenetic effects onto various tissues, and this may reduce the chance of conception, but there is no direct evidence of the mechanisms there.
We do have one more question.
The placenta is protective against virus, but the recent report from Wuhan revealed COVID-positive antibodies in the blood of neonates. How could that be possible? Yes, so normally what happens is that the placenta does not allow the virus to go. The placenta does not allow IgM to go, it allows IgG to go. So what happens is, if the mother develops IgG antibodies, there are receptors on the placenta and the antibodies can go. So placental transmission of antibodies is seen. The virus somehow, most infections in the mother, the babies are protected because of presence of placenta. Now, in this case, what happens is that the placenta has the machinery itself to get infected with the virus, so the virus can transmit here. Now, once the virus transmits, the mother also sends antibodies along with that, so that the virus can be cleared from fetal circulation. I hope that answers the question.
Yeah.
Okay.
So any more questions? Maybe we'll wait for a minute more. I see a lot of young minds deeply thinking. You've really stimulated their thought process.
I hope everyone has enjoyed the talk.
Absolutely.
Slightly heavy on the research side.
But you made it so lucid. It was quite easy to understand, and suddenly out of the blue, we felt that research was quite easy, which obviously it is not.
No, it is. It is easy. It's just that you need a little bit of perspective. That's it. So I love this quote again, which says that discovery is all about seeing what everyone else has seen, but thinking what no one else has done.
Beautiful.
So, in IVF, why are there multiple fetuses? There is a better person to answer than me. Sir, I would want you to answer.
Now, that's brought down to a large extent by transferring blastocyst. Now, the present protocol says if they go up to the level of blastocyst, we transfer only one, which cuts down a maximum 2. Earlier days, it used to be day 3 transfers, and the quality of the embryo were not that good, so we used to transfer more than 3 to 4 at a time, especially depending on the age of the patient. But now it's been brought down, and it is only 1 or 2.
Yes, so multifetal gestation essentially is because we used to transfer, as I said, we used to transfer a lot of embryos to begin with. But now with the success of IVF getting better and better, now I think I'm sure sir practices only single embryo transfers. So it reduces the chance of multifetal gestation. And that's one of the risk factors of IVF.
Excellent tributes pouring in, Dr. Deepak, for your talk. So you can see that firsthand. So, yeah. So if there are no more questions, I think we can wrap the session up until we meet Dr. Deepak with one more beautiful talk. Thank you so much, Dr. Deepak. You're really a busy person, and you've taken the time off to be with us and talk to our students and other delegates, other professionals, doctors, everybody who was online. Thank you so much. It's a great honor to have had you this evening. And on behalf of our institution, we extend our gratitude. Thank you so much.
Thank you. Thank you so much. Thank you, everyone.
Thanks.
Thank you, Dr. Cherian, sir, for your time also. You've gracefully accepted to moderate this session, and you've done an admirable job. Thank you so much, sir, for coming over and moderating this session. So with that note, until we come back with another impactful session, we sign off from Believers Church Medical College and Hospital. Thank you so much, everybody. Thank you for this wonderful evening, Dr. Deepak and everybody. Thank you so much.
Thank you. Thank you, everyone. Have a nice evening ahead. Bye-bye.
Thank you, Will. Thank you. Thanks to everyone.